data table and analysis. So we'll go through that just to try to help you out in time a little bit at the end of it. Okay, I kept the points the same, so and we'll talk about that. So now, before we start, are there any questions on how things are going, or what 2 things you have left to do before I finish the notes?
Mr. McCreary.
Yeah.
So I accidentally started the test because I thought we were taking it in class today.
Oops.
So, could you like reset it, please?
Did you get me a Christmas gift? No? Can you hear me? Yes, I'll reset it for you. Don't worry.
Thank you.
I'm not 100% sure how to do that without doing it for everybody. So nobody take the test until, because I have off between 9 and 10. No one take the test till at least 10 o'clock, because I'll need some time to conference with them. All right? We good?
Yeah. Thank you.
No problem. All right. Are we ready? So I'm going to share the screen with you. Please let me know you can see it. Somebody speak up. Shake your head, Addison. You see it? Okay, I see a man shaking your head. Okay, so we talked a lot about surface area to volume ratio in lungs and alveoli in humans. We talked about surface area to volume ratio, which is an ongoing battle for living systems to get as much surface area as it can for the volume. We talked about the simple graph of how that works. We talked about alveoli and how important they are. To move oxygen into the system as well as moving waste products such as CO2 out. We talked about the harmful effects of vaping and cigarettes. We talked about what does this membrane look like. A reminder that this is a fluid. Notice it says fluid mosaic model, which means it's literally a liquid, which is very difficult to think about, but this is a fluid. So it looks very solid, but if you would touch this, it would just kind of move almost like basically like a fluid, like water. But it's made of that phospholipid bilayer embedded with proteins, cholesterols, and the carbohydrates. So you need to know how this works, and that's in our review sheet, and we'll talk about that when we get to it. But you need to know this slide. You need to understand that this whole area is what controls what goes in and out of the cell. That's the hydrophobic area. The red dots or the phosphate heads are your hydrophilic area. Okay. We talked about all the different types of proteins and what they do. We talked about some of the effects and some of the cool things that happen in the world because of how these are genetic protein that are moved through populations. And then a really important slide here, we talked about things that can go in and out of a membrane are always uncharged and or small, or things that have a charge, like a chloride, which would show as a negative, or a sodium, which show as a positive, cannot go through the membrane without either a channel protein or a carrier protein. Large molecules such as glucose, same situation. They can only go through if there's a carrier protein. Okay, you need to know your passive transport, diffusion, osmosis, facilitated diffusion, with a focus on that part of the definition. It says net and random. The idea that molecules and atoms are constantly moving randomly. You cannot predict where they would go. You can predict where the net movement will go. So the net movement would be usually or always from a high concentration to a low concentration. And we'll talk a little bit about that idea of net in just a minute. We talked about the different types of passive transport, especially in this case, the carrier mediated facilitated diffusion and the channel mediated facilitated diffusion. And then this guy is the one that's causing a lot of problems, and I'm going to help you on the test question on that. So make sure you have a piece of paper in front of you. because the test question is actually harder to understand than this one. I want to make sure we're ready for that. Okay. Let's do it now. You guys ready to do it right now since it's there? Let's do it right now. I need a little extra sip of coffee for that one. So I'm gonna draw. Let me move some stuff on. Is everybody ready? Show me your pencils and pens so I know you're actually writing the thing. I know you're not children here, but this is big, so you better be ready to go. Alyssa, are you ready to go? Draper, you feeling it? You feeling it? Okay. So if I have a beaker and I have what's called a selectively permeable membrane, which is just fancy word for saying a membrane, right? So when I say selectively permeable, that means that certain things cannot go through, usually large or charge. So this shows the fluid level. Okay. And by the way, this is a classic ACT question. You'll see the stuff like this all the time because they want you to think backwards in what happened previously. So let's say that the fluid levels are uneven. So you're going to have this picture, and it's going to say, predict what this would look like before, assuming that the levels were at the same height. So we kind of do this. This is before, and this is after. And we have an SPM, and we have our levels at the exact same height. So assuming that there's something in there besides just water, how could this happen? And this is a really tough question. Let's just talk about the basics. What happened to make this situation? What must have happened? What must have moved?
Water.
Water had to have moved. Had to have moved. So we know water moved into this direction. Now, what's really hard about this compared to this slide, I'm going the wrong way, sorry. Compared to this slide, is it's not showing you the water or the solids. So you're just trying to kind of figure it out based on what you can't see, and that's not unusual. So what must have been, and we're going to say this is the right side, and this is the left, so we can be clear on what we're saying. What must be true on the right side as far as solutes or sugar goes? What must have been over there?
There's more like solutes on the right one than in the
Okay.
left.
So let's say I'm going to let's just put 8 of them in there. So I'm going to give myself a key. I'm going to say that's a solute. Okay, when I say it's a solute, that means it could be a sugar, that could be whatever you want. But what's important about this to help you remember is these guys cannot go through the membrane. If they could, they would just move right across that membrane and then balance themselves out. That would be the net random movement, but they can't. So because of that, let's say that there was 100% water solution over here with no solutes. And let's say that this was an 80% water and 20% solutes. So yes, the amount of solutes, because this would be 0% solutes, right? The solutes would move, but they can't. So what you do next is you look at the water movement. So the higher concentration of water is on this side, the lower concentration is on this side. So the water will move in this direction. And that's why the fluid level is increased. Give me a thumbs up or a happy face. I'll give you a choice if that kind of makes sense. Good. Alex does good happy faces. I'm going to give him extra credit for doing a good happy face. All right. So now, when we're looking at that, here's the hard questions, okay? So listen very carefully what I'm going to say, because this is a little bit tricky. I'm going to ask you 2 different things on this test. One, I'm going to ask you for a term called the amount of solids. And then the second thing I'm going to ask you about is the percentage. or concentration. You can use either of those. So when I say amount, if I look on the right side, how many solutes are on the right side?
8.
8. So if they can't move across the membrane, how many solutes could you predict over on the before picture? They can't move across the membrane, so
Wait.
yeah, it'd be 8. Right? It'd be 8. Okay. The amount of solutes can't change. Now let's look at the right side here and compare it to the right side here. Will the percentage or the concentration change? Because I added more water, right? So would the concentration change?
Yes.
Yes, it's going to change. It's like if I have a nice cup of iced tea that's made perfectly, and somebody comes around and pours tons of water in it, will it taste different? Yeah, it's less concentrate. So that is what you have to really pay attention to on this test. There's a difference between saying the amount of solutes and the percentage or concentration of the solution. So you have to be very careful. You have to be a very good reader on these tests. And that's why ACT likes these, because they make you think. So they're going to ask you, did the amount of solutes change? Yes, it did. Did the concentration change? Yes, it did. Okay, so the solute or the amount did not change. This did change. Okay. Is everyone able to see the difference? So that seems easy enough, right? But I'm just going to remind you, this is all you'll get. So all the drawings that I made it look easy, didn't it? Made it look easy. But you can make the same deductions by looking at this and kind of understanding what would happen. So this is all you get. That before something, the before picture is what I drew. So you're taking this at home. So use your notes, right? Or draw it. You draw what you see. Are there any questions on that? There's about 4 questions on that on your test. And I can't see hands raised. Well, it won't ask you the number of dots. It'll simply say, did the amount of solids change? And your answer would be no. Yeah, so it won't ask you how many. That's a great question, though. Okay? We good? Bella, you like? Brady, you like? Anybody else? I mean, Emily, you like? Okay. All right. Let's talk about this slide. Let's not worry about the picture. Okay, let's just worry about the words. So in passive transport, we had 3 types. We had diffusion, we had osmosis, and we had facilitated diffusion. And they all operated with a concentration gradient. So let's just talk about what a concentration gradient is for. Okay.
It says you're on the same slide.
So what is it? Do you see a blank screen?
No, I see the same slide.
Do you see a blank screen?
No.
No. Refresh your computer.
Okay.
Tell me when you get it, because I don't want, this is kind of important. Okay. Can you, can anyone not see this? Speak up. If you are trouble seeing this, please speak up. Okay, so we're going to look at the term because this term shows up a couple times and we've talked about it, but a concentration gradient. So if I have a membrane and I have, let's say I have oxygen on one side of the membrane, I have tons of oxygen over here. I have, and we talked about this with the lab, this is a concentration gradient. And a gradient means it's different on one side than the other. So think about a grade. And a grade is, they use it to talk about roads all the time, like the steepness of the road. So if this is the steepness of the grade on the road, there's a difference in height here than there is here, right? We can agree on that. There's a difference in height at 1 as there is to 2. Okay, so here we could say there's a difference in concentration in 1 to 2. Okay, so if it's oxygen and it's small and uncharged, if this is my left side and this is my right side, what will those oxygen molecules do and why?
They will go into the area that has the less concentration.
Good. Now be very careful because each one of these is moving randomly. So they'll randomly bump into each other, but overall, the net movement will be this way. Now be very careful. Does some oxygen move back that way? Yes, because it's a net and it's random. We have no way of knowing what an individual, but the overall net, there's 4 arrows going one way and one small one going the other. So the net movement is there. That's a concentration gradient. Okay. Now sometimes if I have oxygen and I want to move my oxygen and I literally want to go this way, will it happen naturally? Will it move from right to left? Will it happen naturally through diffusion? Somebody say no. Somebody say no. I didn't hear it. Okay, thank you. So what has to happen is you have to get a pump. And this pump uses ATP. It uses energy. And it will literally pump that oxygen to that side. Okay. So this situation would be against the concentration gradient. That would be active transport. Okay? You know, I got to go let people in. Everyone's getting booted out today. Is Comcast going on strike or something? What's going on? Okay. Everybody back in? Okay. So we have to be very careful on the test. Listen very carefully to this. There are situations where I have tons of oxygen on one side and I have a pump and it'll literally still pump it this way using energy. So this question on the test is the most missed question of all the questions I have, but it's a good question. So let's look at the slide. So you notice it says active transport uses energy, but can be with, which is what I drew here, can be with or against the concentration gradient, which is that I drew here. So it's independent. It functions totally independent. And I want you to think about this. Think about a pump. Think about a sump pump. Usually if your basement floods, you set up a sump pump and you pump the water out, right? Could you, if you use the same amount of energy, could you actually pump water into your basement? Yeah. Doesn't matter, does it? So active transport can pump in any directions. It's independent of the concentration gradient. Okay? That's a very often missed question. Let's make sure we get that right. Okay. Do we know isotonic, hypertonic, and hypo? Are we comfortable with that?
Mm-hmm.
Okay. we understand what will happen to cells when they're under different pressures. Like this is under high trigger pressure because of a hypotonic. That means the water is racing in. Okay. And we understand that water is going to go in and out in an isotonic. And then in a hypertonic, which is your corn syrup or your syrup, the water raced out through osmosis. Okay.
You and Mom got weirdo friends. She always would give Mike, was it
All right, we're going to make sure everybody's muted.
Mary on your rides and they were walking him and all the kids?
Are we mute everybody? All right, there we go. Okay, back to business. You ready to make it harder? So we talked about this equation, right? Did we talk about this equation? We talked about how surface area, the distance diffused, and the particle pressure are all really important. So when I have particle pressure, if I have tons of molecules over here, this is what they mean by particle pressure. Let's say I had 80 and I had 4 over here. What would my change in particle pressure be? If I have 80 and 4, what would the change in particle pressure be? You guys there? What would my change in particle pressure be?
76.
70. Thank you, Alex. You're my favorite student right now. Okay. So we understand we could calculate the D. We understand the A. This is our lab. So I think we're in good shape there. Let's talk about fish. So with fish, there's only one question on the test, and I want to make sure I cover it. Normally, we build a gill in this class, but for reasons not of our fault, we didn't get to do that. And I just want to make sure you understand that when we take in oxygen as humans or as an animal on the land, we take in oxygen and we take oxygen in and filter the oxygen in, even though there's more nitrogen and more carbon dioxide in the air. We're taking that oxygen in and we're moving it into our bodies. Fish do the same thing. So you're going to have H2O everywhere, water everywhere, but you're also going to have dissolved oxygen in that water. So as that water with the oxygen goes in, it's created a system, just like alveoli did, that the gill is all folded into small little parts to increase the surface area, then that oxygen is going to move into that bloodstream. So the idea that we have to understand is that all animals try to maximize surface area so they can get that oxygen into their bodies so they can respire. And that's the basic idea of this slide. All right, we're going to skip that one. Any questions? Okay. Any questions? You guys feeling good about this test? I do. I feel real good about it. I think you're going to totally rock it. All right. One of the last things we'll do is we're going to go through carefully our review sheet. Okay. So this should have been sent to you. Did you guys get this? Somebody give me a smiley face so I know you. Did you get this? Okay. So I'm trying to get you guys to study differently. And I think part of the problem is that you guys work really hard to memorize things. Well, right now it's a take-home test, so who cares about memorizing? That's not how you study for a biology test. You don't memorize. And I think that might be part of the problem. You have to understand it. So there's a couple things you can do differently. Number one, you could try a different study method. And I'm telling you, even though you think this is a take-home test, you still have to study. Okay, one way you can do it is by using a concept map. And a concept map is basically where you take your notes that are on in your notebook or on the computer, and you literally reorganize them into your own brain. So you take a big subject like maybe passive transport, and then you break that down into like maybe the smaller ideas. Could be diffusion, osmosis, carrier mediated facilitated diffusion, and channel mediated carrier diffusion. And then you break all those ideas down and show examples. And you're reorganizing in your writing. There's tremendous research out there that says that when students write things down and reorganize them into their brain, they remember them and they learn them. Very different than just looking and trying to memorize. The second thing you can do, and I think this is very helpful, is simply paraphrasing it. So you could write your notes down and then literally just make little boxes around your notes and just write little paraphrasing statements. So when we did the active transport, you would just kind of make a little box in that passive transport area of your notes and simply write down a summative sentence. Like maybe you would write, this always goes with or against, it's independent of the concentration gradient, just to kind of get it into your brain and write it down. Okay, that's called paraphrasing. Just looking at something and just kind of getting a general idea of what it is. I also like figuring it out. It does say textbook. We don't use one, so you'd use your PowerPoint. And in which case, you'd literally go and only focus on pictures. And remember how we wrote captions in our lab? You'd basically be captioning all the pictures. Okay, what's going on in these pictures and try to work on that. All right, next. Is the simple outline. This is forcing your brain to reorganize that information. So we can do that. Okay, and then I would not do Cornell notes right now. I think they're too complex to learn on such short notice. So I put that in there for you to have, but I would skip that for now. All right, let's get to the content. Any questions on those? Okay. So we're gonna go step by step. Make sure we understand this. Understand how a membrane is fluid. Notice I've mentioned that fluid part 3 times, so it must be important. Consisting of phospholipids, proteins, carbs, and cholesterol. You have to understand the idea of selective permeability. It's kind of like a bouncer in front of an establishment. They let some people in, they don't let everyone in. People or the molecules they don't let in are large and charged particles. Everything else can go through. Okay, 3, understand passive transport, specifically diffusion, the 2 types of facilitated diffusion and osmosis. Make sure you know the terminology on these. 4, understand concentration gradients and why they form. Well, the why they form is kind of hard, but remember, you have to have something that won't pass through a membrane to create that concentration gradient for osmosis. For example, you had to have sugar in the water on one side to make a concentration gradient. Now, you have to have more molecules on one side than the other to make this happen. How did we do that in our lab? How did we create a concentration gradient so water could move? Somebody answer me that. How do we create that? This is on the second day of the lab. What did we do?
There was more sugar in the syrup than the egg.
Right, and that's your concentration gradient. That's what forced the water across. Good job. All right. Understand different ways life utilizes the concept of surface area to volume ratio. I'm thinking alveoli. I'm thinking about plant roots, how they send out those tiny little roots to get water. I'm thinking about gills. All those things are way life actually does this. 6, given an example of selective permeability, selectively permeable membranes and concentration gradients, be able to predict osmosis, be able to distinguish between amounts of solutes and concentration of solutes. That is describing the picture that I drew for you, the before and after picture. Okay? I think we covered that pretty well. 7, understand why the terms random and net are used in diffusion. Make sure you understand that these molecules are constantly moving in a random manner, but there could be net movement if there's a higher concentration on one side than the other. And again, we talked about active transport. It can go with or against. So you can pump things against the gradient, or you can pump things with the gradient. Now, you need to know what an alveoli is, or alveoli is singular. You need to know your terms iso, hypo, and hypertonic solution. Be able to match them up on a picture, which you've seen in your notes. I'm not trying to trick you. I just want you to know it. And you should be able to match up all of your passive transport, carrier mediated, channel mediated, and regular diffusion and osmosis. Match those up. And then finally, 12, I'm going to give you, remember in your winter survival test where I gave you the A and I gave you different animals and what they're doing, and it was like a data table and you had to calculate the Q. You remember having to do that on your test? It's going to be the exact same thing, except I'm going to have different animals and you're going to have to calculate the rate of diffusion and answer questions on that. So you're going to need to have a calculator handy. Okay. I think everyone knows how to calculate that rate with the particle pressures, and you need to make sure you understand how the equation works. Okay. Let's see. Make sure you understand all the parts of the selectively permeable membrane. And then there's a situation where I give you a graph and I have you read the graph, and it's nothing that you've ever seen before in our class. And basically, you have to be able to look at the y-axis, look at the x-axis, and then decide if this graph supports or rejects the given hypothesis. Okay, and that's that. That's your test. Mortals, do you have any questions? We went fast, but we've been kind of going over that same stuff. This is like the third time we've covered some of the same thing. So I'm feeling good. Okay, please get your faces closer and give me a degree of smile on how confident you are on this test. If you're really confident, a huge smile. If you're not very confident, a frown. If you're kind of mediocre, a smirk. This is a smile. Addison, what are you doing? Is that a smile? Yeah, that's okay. So we're feeling pretty good. Okay, I like it. I like it. Any questions so far?
Over what we're supposed to have for the data table for the lab.
Okay, let's do the lab. Before we go on, are there any questions on the test? Do you understand how to take the test? You're going to log on, use your password and username that I sent you. If you're like, I can't find the test, I only see history tests, that probably means you use your history login. So use your science login and you will see a science test. You have, I think I set it up for today all the way to the beginning of Friday. That way you can take it and decide if you want to get rid of it or not. Right, Trevor? Okay. So that will take care of one thing. Really quick before I go into the lab, I don't want to forget to tell you, how many of you had trouble with the coloring project getting a good grade on that? Or not getting an accurate grade? Because some people, what you're doing is you're using the PowerPoint, and you're turning in the PowerPoint with nothing on it, and then sending me a picture afterwards of taking a picture of the PowerPoint with the coloring on it. If I get something in the grade book, one thing shows up, the grade. And it's usually the blank document. So I in turn give you a 0 and I'll write nothing there. If you got that comment back and you actually did it and you want the points, please message me and tell me to go back through and check it again. Because a lot of you are turning in blank work first, and that's what I grade. I can't go back through and grade all 85 documents that some of you guys sent. So tell me, we'll correct it, we'll take care of it. Don't tell me Friday, because I'm going to be grading labs and tests. So tell me as soon as you can, okay? Just send me a chat. All right, lab, what was the question?
For the data we're supposed to have in our data table and like how to set it up.
Okay, so I gave you a model data table. You are not turning in that sheet to me, okay, that I sent to you. You are going to make your own data table. Now, it's very simple. You just have to model after the one that I gave you. So I think I asked you for fluid amounts. I asked you for some very simple things. If it's on the sheet I gave you, you just have to mimic that and make your own data table. Nothing more, nothing less. Okay, just the things that I asked you to get. The hard part on this is some of you really struggle with coming up with a title. Your title cannot be egg data table. You have to think a little bit more about this. It's a summary of what you collected. So you have to think a little bit about this title. Data table is a bad title. Egg data table is a bad title. Egg and syrup is a bad title. You have to think about what's going on and name it and what's going on, right? Okay. Any
Is it
questions on?
okay if we have multiple data tables?
Well, I think on my sample I gave you 2, right? Yeah, so I would imagine you wouldn't want to have any more than 2 or 3, and then they each one should have a title. But yeah, that's fine because you also you want to have basically the information you collected. But what I gave you should be sufficient. Just kind of make it your own. Okay. You still have to label it results. Put the data table in there, and then you're going to write an analysis. I hope you guys are writing this down. Analysis should be labeled, right? So the analysis, there's 70 points in your analysis. So maybe we should talk about that a little bit, right? Maybe. Okay. So an analysis is made out of 3 things. What are they?
Evidence reasoning.
Good. So the claim is going to be you either support your hypothesis and clarify that or reject it. Do not say prove. If you say prove, I'm done reading it. You're going to get a 0 on that part. Please don't say prove. Don't say found it true. You did not find it true. Okay. All you did is you either found data to support it or reject the claim. Those are the 2 options. Or not enough data to support or reject the claim. Don't say prove. Everybody unmic yourself and say that.
Don't say
Say proof.
Approved.
Okay. If you guys are going to fake it, you at least have to lean forward and then mouth it. Okay. Next time. All right. Got it? So now we're going to work on the analysis part. That's the hardest part. That's your evidence. What evidence do we have? And maybe we should pull this up so we can kind of see it. Give me 1 second. I want to make sure that we're doing this right. Because nothing would make me sadder if you got a poor score on this. Patience. I'm doing technical things. I'm not quite sure what I'm doing. Okay. Everybody see it okay now? Great, thank you. So when we look at the analysis, claim or support, rate of diffusion has proper unit. Did we talk about the proper unit, what that proper unit is and how to derive it?
Yes.
Okay. So we know it's going to be a cube per second, right? A centimeter cube per second. Okay. So you need to make sure you have that. Evidence clear, relevant, and thorough. So the evidence is going to be your rate of diffusion. And can we just have one person say a sentence on how they would use that? Because I get a lot, people get confused. They're like, okay, I'm just going to say that the evidence and the data supports my hypothesis, which is not what I'm looking for. I want a actual number. So let's just practice. What number would you use to support your hypothesis?
Rate of diffusion.
Okay. So let's say, I'm just going to make up a number. Let's say it's 100,000 centimeters cubed per second. Okay. So let's just talk about that. What does that number mean? What does that number actually mean? Sorry, I have to keep bringing people back in. What actually is moving?
That there's water particles moving
Very
into
good.
the syrup.
Good. So it's the rate of diffusion of water, which is technically osmosis, right? So we have to use that number to support our hypothesis. Now, any questions on that, you have to have that number and you have to know how to use it. The reasoning too, notice it ties the claim and the evidence together. So you have to make a link there. But the hard one is reasoning is well written, concise, and specifically mentions why water movement occurred. Let's just make sure we all understand it. Why did the water move? What did we create? What technical vocab term did we create?
Hypertonic solution.
We created a hypertonic solution, which in turn created a concentration gradient, right? So we created a concentration gradient, which allowed the water to move from a high concentration to a low. So at that point, notice the flows in the next part of the analysis points, where you actually describe how osmosis happened. Okay, that's not easy to do. Osmosis is moving because the concentration of water was higher on one side than the other. Okay. And you also notice that you have to have concentration gradient, and it's explained in your report. It's 100 points. Okay. We don't have time for you to turn the sunlight. We don't have time for you to piddle on this and not do it. So let's make sure we get this done. Okay. Any questions on this? Two grades, people. And you get to get rid of a grade too. I feel really good about it. I think your grade's going to jump. You got 200 points in the next 3 days that you're going to add to your grade. So that's a lot of points. Typically, the score on the plasma membrane test is much higher than the winter survival test. Typically. So I think that should give you a little bit of confidence. Yeah, the winter survival test is a really hard test. We good? Alex, what are you drinking?
Hot chocolate.
Good. Okay. We good, guys? I'll be on the chat. I'll be responding to you. I'll be listening to you. So if you need help, use that chat option and we'll go from there. Other than that, pretend you're snuggling up with a blanket and you can log out. How come you're not snuggling with blankets?